Okay, can you see that?
Yep, looks great.
Great. Sorry. Yeah, so thank you. I'm Paramvir Dehal. I'm going to talk about KBase and its use for integrating multi-omics data for sharing, analysis, and modeling. So one of the things that we're trying to do or trying to facilitate is mechanistic modeling and validation in KBase. And so what we try and do is starting with genomes and genome annotations. We try and annotate the genomes, build species-specific models, and then try and move towards more community models and connect our sort of models of isolates and individual genomes to the ecosystem. And so for a lot of this, what we're doing is generating these flux balance models and then trying to gauge whether or not they're accurately predicting what's happening in the environment based on experiments. So we have growth phenotype data, or KBase can process this growth phenotype data, gene expression, proteomics, and metabolomics, and use that to reconcile these models and try and adjust the annotations, make these improvements, and then propagate these improvements across, basically across the tree of life. So one of the things that we're doing is integrating these flux networks and pathway predictions where we have these genome scale models. We're able to identify the metabolites that they produce. Some of these metabolites are consumed to generate biomass. We can map them to existing biochemistry data sets, and then we have some that are effectively just unknown and not part of reactions. So these are sort of missing reactions that we don't have captured. And one of the things that we're interested in is whether or not we can use metabolomics data to help improve our models. So one initial effort that we have has been with Trent Northern, who spoke on Monday. And one of the things he's been doing is generating this exome metabolite data. And this process, so what is exome metabolite data? It's effectively, we start with rich media and quantify that with mass spec, then inoculate it with your microbe of interest. And then after some period of growth, re-quantify the media with mass spec, and then just compare the metabolite abundances before and after. The assumption being that the preferred nutrients are going to go down and the byproduct metabolites will go up. And we can use these exome metabolites to fit our models, predict new genes, predicting genes to perform new pathways, and then connecting these results to ecology. And there's already 10 SFAs that are kind of using this this tool set that Trent offers through JGI. And it's an important tool for understanding microbial physiology. So we've loaded a large amount of exometabolomics data from Trent's Web of Microbes org into KBase. So we have one central narrative containing the full data set that WOM offers. There's over 589 exometabolites observed across 56 conditions and six microbial species. So what we've done is you can observe the raw data through KBase, and then we've mapped all of the compounds with model seed IDs, and this is what allows us to build the models. So our narratives include the isolate genomes, the annotation, and these reconciled models. So this reconciliation process is relatively complex, but the basic workflow is that for genomes that don't have existing published models, They are imported into KBase, annotated, and then we build a genome scale model. For the genomes that do have published models, we import the published model. And then for the Senecacoccus genome, instead of building the model, what we do is we use a published model from a very closely related species, and we do a proteome comparison to map the model from effectively one Senecacystis to the Senecacoccus genome. And then those models we use in our exome metabolite analysis, the apps that we have in KBase, and that produces, that allows us to reconcile it with the flux balance analysis and then gap fill the reactions. So what we get out of this are predictions of gene candidates for these gap filled reactions. And so those end up being new annotations that can be propagated across our system. So the apps, like how does this work in KBase? Like, so we've added 2 additional apps to, we've added this additional app to KBase. The user specifies their model, base model, and the exome metabolite data set. And then we can automatically gap fill that model to see what metabolites are being consumed and which ones are being produced. And then there's a report. So when we try and do this fitting, the app predicts what reactions need to be added. Some reactions, like here, adenosine just worked, so it didn't need to have additional reactions added. So the workflow behind the scenes is effectively we filter the exome metabolites that weren't found in the Model C database. We create transport reactions for the metabolites in the model, and then gap fill the reactions based on the Model C database to permit the simultaneous use of these inputs and the productions of the output that we observed. And then this allows us to create this new media with successfully gap-filled input and output exome metabolites. And then we save these reconciled models and identify the missing functions. So there's some identifying the missing genes is the next step. So when we try and find these gene candidates, we've identified gene candidates for about 45% of our gap-filled reactions. And then these exome metabolite methods, we found 93% of the exome metabolites were in our Model C database. And so based on this analysis of just 6 isolates, we have predicted genes for over 80 missing functions. And so these predictions can then be used for the rest of, or for improving the rest of our KBase isolates. So our goal is to really kind of connect all of this to the ecology, to what's happening in the environment. And I've talked a lot about like the metabolomics side, but we're also trying to include additional experiment information like growth phenotypes, gene expression, and proteomics. But what we find is that the gap that we have ends up being connecting all of the data together. So you can kind of do a lot of this in KBase right now, connecting proteomics and gene expression and metabolomics and improving these models, but it ends up being a lot of like very tedious by hand exercises where it's difficult to find the data that you need to do these connections. So what we've been doing, and we've just released this new functionality in KBase, is a new sample service. And that allows us to track all of the metadata related to a sample, as well as the experimental data that was generated from that sample. So metabolomics, proteomics, expression data, all gets linked to sample metadata. And then we can have a more straightforward integrated analysis that allows you to combine all of these multiomics data sets in our KBase narratives. So another reason why we're interested in samples is this is where we see as a path to this exploration and discovery that we want people to do with KBase. So capturing sample data is key to enabling this fair, findable, accessible, interoperable, and reusable data. Users can locate samples based on some straightforward search interfaces like geolocation and the linked data types. So if you have amplicon, genome assemblies, metabolic models, chemical abundance, you can find it based on the data that's been generated from those samples, and then see this, ultimately how other users have used that data. And the power of the samples will increase as we kind of get more of it in and we start generating more samples from users and bringing it in from other sources as well. So we've kind of thought extensively about samples and how to define samples. And we're taking a very kind of practical approach at this point. We're trying to collaborate with other organizations to converge on standards and definitions, but from a very operational standpoint, we consider a sample to be a physical material collected from some volume at some certain time. And we allow users to attach attributes using ontologies and other defined templates. And then we can use that to connect measurements that were made against that sample or measurements against the environment that it was found in. And this is mostly to facilitate data organization and enable the analysis that we're interested in. So for the properties of these samples, users can define these, they can use controlled terms to assign attributes, and they can also have user-defined attributes. Both types of attributes can be used in analysis. The controlled attributes are conceptually very well defined and they're standardized across all of the samples. The user attributes right now are not currently validated, but we are kind of moving towards improved validation for user types. And because some of these are user defined, they're not guaranteed to be consistent across samples. And that's another area where we're trying to improve things so that we can support things like meta-analysis across multiple data sets across different labs. We are focusing on generating templates that help us translate these spreadsheets into this internal data representation, and the samples can be linked to the raw data. So we're kind of improving, we're trying to focus on using ontologies so that it's easier to understand the content or the attributes themselves. Right now, we're in our sort of 1.0 release, we've been focusing on subsurface data and primarily collaborating with ESS Dive and using CSAR formatted IGSN linked samples and the Enigma SFA templates. And so the validation includes types, range, and ontology validation. We can link reads, amplicons, and chemical abundance right now. We're kind of expanding that in our next releases. We've added the viewers, the tools for uploading and identifying errors in upload, and we've generated the apps and analysis tools so that users can do this analysis of subsurface amplicon abundance and chemical abundance data sets. So right now, like I said, this is our sort of 1.0 release. We're still working to kind of expand more functionality. Right now, you can use this to organize your data, compile metadata, inspect the contents, and then perform these correlations of things like metabolomics and amplicons. So like I said, this is just the first step. We're still trying to move towards capturing experiments and protocols. And we're trying to make sure our samples framework is interoperable with other sources like ESS Dive, NCBI Biosample, EBI, NMDC, JGI, and EMSL. And so ideally, we want to enable linkages across these systems to find relevant data. Whoops. Sorry, those two were repeated. So samples and chemical abundance, this is what we're trying to do is connect chemical abundance data with samples, and then in this context, things like the exome metabolites. So we have a number of use cases, and we've got some narratives that we've built using these this sort of beta functionality with the apps that allow users to visualize the metabolomics in interactive Escher maps. There's an example here. Reconcile the models with the exome metabolite data using the same methodology we use for web of microbes, and then correlating the metabolomics with individual OTUs. And so this is an example of some of our metabolomics data from Enigma. And the import process is relatively straightforward. It does require that the users have to generate all of the metadata, all of the specify the sample format, the template, and then they can be brought into the system and shared. And the sharing follows all of our sharing models, so things can be brought in privately, shared with a select set of users, or shared publicly. So there's some template generation. And then we do all of the error checking and making the links with the existing data across the system, so that when you start doing your exploration in the narrative, you can then get some basic stats on your chemical abundance data, and then follow the kind of exometabolite processing that we've seen in the other web of microbes narrative. So our upcoming, so this is a new kind of functionality in KBase, and so we have a series of webinars that are associated with samples. So we've, coming up next is just the tutorials on how to create and manage the samples. And then we'll have a larger, longer tutorial on how to link or how to process amplicon analysis in KBase, and then how to link chemical abundance and amplicon analysis, and use these to build community metabolic models. And then finally, there's an additional webinar on linking multiple forms of measurements and then performing correlation analysis across them. And so with that, I'd like to acknowledge the software developers on KBase, the KBase staff who've been working on this. We've got a series of software developers, user interface developers, the subject matter experts who've been driving a lot of the amplicon and the amplicon and chemical abundance analysis have been Pamela Weisenhorn and Janneke Erdősing, both at Argonne National Lab. And I'd also like to acknowledge our PIs. Chris Henry has been doing a lot of our functional genome or metabolic modeling work, and Adam has been facilitating all of our subsurface data analysis. So thanks.
Thank you so much, Dr. Dehal. I think all the work that KBase is doing is really exciting. And I mean, I'm just personally impressed that you managed to get a cat working for you. They're usually not that great on software developing. There's a question in the chat. Will KBase be connected somehow to Algebase, or what efforts and when will that be done to bind the data together?
So we're trying to facilitate like some large scale kind of data imports and have things that we kind of consider these data compendia that can be used by kind of KBase staff. So we're trying to import data from, like we did with Web of Microbes, of bringing it in, kind of standardize it and make it easier to process and share and use in your own analysis. And so we haven't started kind of looking out at a lot of these other data sources like Algebase. But those are things that we would want to target as data compendia to bring into KBase and just kind of make it easier for users to use it in our existing narratives.
Awesome. Do you have a standard sample process that you could recommend that minimizes the degradation of the sample when exposed to light and oxygen?
We don't make, so this is the thing with KBase is we don't really make a lot of recommendations on like how to generate the data. What we're trying to do is capture how others have been processing the data, you know, the protocols that they use. And then, you know, our concept is more that if we have a lot of variation, a lot of data using different methods, users will be able to kind of sort that out and figure out which methods are kind of working best. And so for some communities, that may be one particular set of protocols, but for another community with their environment, they may have some other set. So we're not prescriptive in, you know, protocols or how it is generated. We just are trying to kind of capture all of it and make sure users are aware of like how the data was generated.
Yeah, awesome. Well, I want to thank you once again and then extend a huge thank you to all of our presenters this morning. Please stick around. At about 1 o'clock is my tutorial on high performance computing at EMSL with some data from Neon. And that begins at 1 again. And the tutorial will take place in a separate Zoom meeting. The link is in the chat. I'm going to turn the floor over to one of our co-chairs, Eric Choi, to close out the meeting.
Oh, thank you, Allison. I'd like to take this opportunity to thank everyone for attending this year's EMSL Integration meeting. I'd also like to thank all the speakers for the 4 days of fantastic presentations and the great posters I saw. Also want to remind you that the meeting recordings will be available on the EMSL Learn website. Recordings from the previous day are already posted, and we will post presentations from today in the coming days. There's a link at the EMSL Learn website in the chat window now. Please go take a look and you'll find the other videos. I'd also like to invite you to save the date for next year's EMSL Integration meeting. It'll be during early October 2022. Next year's meeting will focus on machine learning and artificial intelligence. Additional details will be shared in the coming months. And once again, thank you to everyone who made this year's meeting possible, and we'll see you next year.